So first of all, I just want to thank all of the speakers who I really think did an exceptional job today in presenting clearly and in a very didactic way to probably quite a diverse audience. I really felt it was an exceptionally high quality series of talks, and as I didn't speak, I'm allowed to say that. And so now we have to conclude. I have to admit, coming after Eva Jablonka, it's quite difficult to conclude because she does cover, I think, many of the concepts and ideas that have been discussed today. François is supposed to help me, but he's sitting down. That's okay. So I put together just a few slides very briefly, and then a list of questions, and then I think people can ask questions, or the speakers, I think, is more to the point, and we can have a debate if you like, if there's any more energy to debate. So here I just thought I would show you the age-old chip off the old block, which I hope we all agree probably doesn't work this way. A poor father who lost his leg somehow transmits it to his offspring. I think we all agree that that's probably not true and not genetic, let alone epigenetic. However, this concept that maybe we can transmit from one generation to the next a certain amount of information that isn't DNA sequence based, I think we're all convinced today that this does happen, it does exist. And what this corresponds to, I think, is one of the big questions for the future. So I just want to very quickly summarize, I think, some of the things that were mentioned along the way, but I do want to emphasize that most environmental changes that we're subjected to are erased at every generation. Otherwise, can you imagine? Indeed, we would have, legs cut off and, strange-looking skin. And so that has to happen. And most organisms, all organisms do that to different extents. But as it says here, different organisms do this to different extents. And I think the example of plants, where clearly there are some epimutations that can be transmitted, is very tempting to cite. But in fact, as I think we all understood, so far we don't know of any environmentally induced changes in plants that really seem to be transmitted stably. There are stable epialleles, but they don't seem to be, or we don't know what their environmental induction was. Whereas in worms, I'm convinced now at least, there clearly is very good evidence for this. Oops, oh well, there you go. That was the end of that slide. So here I was trying to say, it's like a cloud, I guess, yeah. What information is transmitted? So I think we've discussed many of the different potential epigenetic marks, DNA methylation, polycomb, non-coding RNAs. We haven't really discussed prions too much, but I think that's something that we have to bear in mind. Proteins can influence proteins structurally, and those can definitely be transmitted. Different organisms have different strategies. One issue that Eva highlighted, that's lost here somewhere, but that I think is very important, is the soma versus the germline. Not everything has to be induced in the germline in order to be submitted. Things can be somatic, and then this concept of reconstruction, which I think is very, very important. This sort of idea of a cascade of events, and you don't have to have any single mark. So those of us who are very happily looking at DNA methylation or at polycomb, I think should be thinking more globally about what changes truly are being transmitted at any particular stage, whether they're in the germline or in the soma. Okay, so I think I'll change slide here. This is awful. As I said, I think in terms of environmentally induced changes, there isn't that much evidence that it can go across many generations. That we can induce changes in the first generation, that a fetus in utero can be exposed to the changes of the mother, whether stress or nutrition. I think we all agree that that can definitely have an impact on gene expression and on phenotype. What the actual epigenetic modifications are, I think we still have to, as I said, bear an open mind. But then, to what extent do those environmentally induced changes go across generations? And we've heard a few people talking about F2 and F3, but people usually start to become a bit nervous when they start talking about F3. It's very difficult in most organisms and models that have been looked at to really be able to dissociate the inducer from the phenotype that is maintained. And in females, when it's female transmission, you have to go at least to the F3 generation. When it comes to the males, F2 is probably enough. I think as I said earlier, in worms, I think there's good evidence that we can go beyond, in flies as well, probably. And in mammals, I think we have to keep an open mind because, well, as in other organisms, but maybe more so in mammals, there are a lot of confounding factors, and the devil is in the details, as they say. Because, indeed, you can never really exclude some DNA sequence differences, and I think we've heard that repetitive elements and transposons are often the targets of epigenetic changes, and epigenetic changes or stress can induce transposon mobility. So when we're all happily looking at our favorite gene that's changed its methylation status, maybe it's because there's a transposon that landed there because of the induced change. So I think that up until now, it was a bit of a black box. But now we can answer this because now we can sequence genomes and it's not that expensive. So I think this field is going to very, very rapidly be clarified because now we're going to be able to look at the individuals that carry the phenotypes that we're studying, that were inherited, and really sort out whether it's epigenetic or DNA sequence variation based. So I think the future is actually very bright for this field because at last people will stop sort of saying, oh, maybe you're not doing your experiment right, or, it's all a bit vague, it's fuzzy. It won't be fuzzy. We will know the DNA sequence and we'll know what the epigenome is and we'll know what the phenotype is. So I'm actually really excited about the future, even though I don't actually work on anything transgeneration, so I can happily tell you all that this is what I think we should do. But the other thing I think that's very important, and that's why I wanted Eva to maybe say a word about the sort of cultural effects. I mean, even in rodents, we've heard about the influence that parents can have on their offspring. So, maternal influence, grooming, we all know that maternal milk is an amazingly rich source of information. No one really talks about that, but, when a mother suckles her young, she's transmitting a lot of stuff. And then I think for many of the rodent experiments, people are now aware that you have to be extremely careful about the way the animals are handled, the kinds of environments we're looking at, the dietary variations. When you buy your chow, is chow always the same? I think people are starting to realize that it's not. So, I'm not criticizing anyone who spoke today because I think the people who spoke today are actually doing the experiments properly because indeed they're aware of these sorts of variations. But I think a lot of the past literature wasn't necessarily aware. So the question in mammals, I think, remains open. In humans in particular, it's very difficult, I think, because of the intrinsic polymorphisms we have and variations in our genomes. Twin studies are not enough when you're looking across generations. And so I actually really like this quotation when people sort of come to me and say, oh, well, epigenetic changes in humans transmitted across generations, where in the end you're putting a lot of responsibility on the shoulder of the parents and the lifestyle the parents have. I actually always do like to talk about culture, and this is Stephen Jay Gould. Human cultural evolution in strong opposition to our biological history is Lamarckian in character. What we learn in one generation we transmit directly by teaching and writing. And I think for me that's probably the most epigenetic one can become when it comes to humans. So it might be indeed premature to compare humans to plants or to other organisms where we can really do the experiments properly. But nevertheless, and I think someone alluded to this, I can't remember if it's Vincent, but in mammals, we're starting to understand now by looking in the germline at what becomes reprogrammed, that there are regions that do resist reprogramming, even though we have to kind of get rid of most environmental changes or even just noise of DNA methylation. We know now from the studies of Serrani and Reich and others that there are true epialleles that can somehow escape this. They're most often associated with repeats. The question is, do they have any phenotypic consequences? And again, I think that's where our capacity now to really look carefully will allow this to be sorted out in the near future. So I think through carefully designed experiments and our capacity to sequence genomes and to look at cohorts of animals or even people thoroughly, we will be able to come to some very clear conclusions. And so I'm not going to say any more. I just want to end with some of the big questions that I think we've covered today that we can ask again or ask other questions. This idea about stability of changes, how prevalent they are, when they happen, also the window of time when these things happen. For example, in utero, clearly different stages of development will have different consequences on the resulting offspring and their generation. So I think I'll stop there. François, is there anything you disagree with? I think we were put here as a duo because we were going to disagree, but actually I think we agreed on most things when we discussed earlier. And I think one thing we agreed on was that we should really talk about genetics, because epigenetics can be distracting when we're talking about transgenerational effects. The question is, can we actually see effects that, sort of follow Mendelian rules? And that's genetics. I think we agree about that, at least. Yes, he's nodding, so.
Few additional words, François?
Well, just a word. I basically agree with most of Edith's remarks. I was wondering, in fact, about the need really to, if there is any more need for the term epigenetic. From what I heard today and what I'm really reading and working on, I think consider epigenetic is just one extension of genetics, no more, no less. And genetic has not to be, it's equated with DNA encoding. It's probably, to me, a restrictive approach. There are many other ways to express a character and to maintain a character.
We have this discussion about genes as well.
Encoding in DNA.
Yeah.
Genetics was alive and flourishing well below DNA was discovered.
That's true, absolutely, yeah.
So there are, the problem with epigenetic is that it often means different things for different people.
Yes, actually.
And,
I thought I'd show this slide. Different things.
Does.
Epigenetics.
Not only epigenetics, but also genetics means different things for different
Well, it shouldn't, but it does.
It does. Sorry, I'm an historian. It does. So if you change the meaning of genetics, then we agree that genetics and epigenetics are the same.
Yeah.
But historically, Historically, I don't think that you can say that after all, what you call epigenetics is part of genetics, because that is what you say today, and I agree with you, but you would not have agreed with me 10 years ago. So, sorry about that. I don't know what you think about
has to, effectively, you're right, I think that we're changing with time, no problem about that.
Sorry about that.
But, no, what I'm concerned is when talking with some people, everything which has to do with chromatin structure, that's epigenetic. I don't know what our colleague Geneviève will agree with that, but I'm convinced that there are changes in chromatin structure that are induced by the regular epigenetic regulatory mechanism, binding of a protein to a sequence initially in the genes, like that. So it turns out to be a bit confusing. It doesn't matter much, probably, because we all know what we are talking
Yeah, I think the important point is to qualify.
Yeah.
To say, when we say epigenetics, say what we mean. Which kind of epigenetics are we talking about?
Then just finish. I had one or two remarks. The one remark I'm struck with is that what we have identified as transgenerational in characters in mammals are only diseases, are only defects in which are we have several examples. I myself gave several in which an abnormal functioning, accidentally abnormal, is maintained. And that's a concern for the medical, the clinical people, of course. But I'm still waiting for transgenerational transmission of a character which would have an adaptative value. I don't know if someone...
Agouti could have.
Agouti
might.
Examples that flowering time can be extremely adaptive, epigenetic changes in flowering time.
Not in mammals.
But you know, this was it was exactly the same. It was exactly the same concern in the beginning of genetics. In the beginning of when Mendelian genetics began to be, people were doing Mendelian genetics. It was exactly the same problem. All the mutations that were found were deleterious. It was white eye, and it was crumpled wings, and people said exactly the same. Maybe it is a superficial kind of characters that are being affected, that are really just deleterious, little deleterious kind of things that are throwing development off the normal trajectory, but things that are really evolutionary important, that give fitness benefits, this we have still to wait and see. It took about 40 years to have good examples in genetics, in natural populations in the 1960s. Endler presented a kind of big review in a book of genetic changes that were beneficial. But it was for a long time, and I think it's natural. I think that's whenever we're studying a new thing, the first thing that we see are the abnormalities, and abnormalities are not good, usually.
I just wanted to make a few comments, in particular with respect to the use of the term, because we have debated on this, and I think that getting back to the history is definitely a very interesting point, because the terms and their meaning evolve with time themselves, and I think we have to consider this when we look at things. So at the moment there's a bit of confusion, because the use of the term is associated with a definition that is not corresponding always to the same timing. So we need to synchronize, and we also need to provide our definition when we speak about a particular problem. I also really liked the point made about replication, and I can say this even more because I was myself very interested by the process of replication and how you duplicate chromatin organization during this time window. But I think the point that is really important is time, and time to reproduce something. And you don't necessarily reproduce this in a manner that is coupled to the event that is disruptive. It can be separated in time. And one example I was just mentioning to Eva is the example of the centromere. At least in mammals, it's not at the time of replication that you make it exactly. There's a delay when you're replenishing things. And there are many things that could be even beyond the time scale of the cell cycle. So I think that we have to be open in terms of where things are reproduced, and also evade from the scheme into which we are usually thinking. So that's definitely an important point.
I have a question as a German. We are used to talk about genetic disease, and we are not so used to talk about epigenetic disease. What is your feeling about that? Yeah, anybody?
I think that the kind of obesity, I mean, if it will turn out that there is a strong transgenerational epigenetic aspect to the obesity epidemics, I think this is a kind of epigenetic disease. I
was thinking about schizophrenia.
Well, I don't know. Schizophrenia is complicated also, but yeah, I think that it is a concept. People today in the plant world I heard a lecture by David Bolcom that was talking about epigenetic engineering. So all the concepts that we know from genetics, genetic engineering, genetic disease, are being transferred to the epigenetic world, especially in cases where we have long-term epigenetic transgenerational effects. And I think we can talk also about quantitative epigenetics, and there are models, there are epigenetic, population epigenetics models and also field studies today. So the field is growing in a way which parallels the growth of genetics in many ways. It's different kind of rules and different logic sometimes, but
maybe, oh, okay. Then I will pass to Jean-François, but now it's your turn.
Yeah, so actually I have a point about environment versus genetics, because people tend to oppose the role of environment or genetically induced epigenetic traits. Like, you can induce a trait with the environment and then look if it is transmitted, or like what we've shown today, you make a modification, a transient genetic modification, and then you induce a phenotype and this is transmitted. And in fact, I have to qualify what I was saying this morning, because we have made experiments that I didn't have time to explain. I'm not going to go in detail through them, but just to say that we have made the same scheme in different conditions. And if we apply the scheme I described to you under temperatures that range in 18 to 21 degrees, which are normal temperature for flies, this all works fine. But actually there's another range of temperature, 25 to 29 degrees, which are equally favorable for flies, in which if we do the same scheme, we do not obtain any epigenetic transmission. And so this means that you can even if you induce genetically a trait, and that is then epigenetically transmitted, this has to occur under environmentally permissive conditions. And I think this is an important point, and I think that you might also have the opposite. When we study environment, we are usually pretty brutal and rough. We don't know how to induce a very specific environmental change to the organism. We can modify each one of our 20,000 or maybe more of our genes, but we have a big trouble to change a very specific environmental set. And so if we were able to induce specific environmental changes under permissive genetic conditions, maybe we would obtain the same. So I would leave it open for the moment.
Yeah, just a few words. What can I add after Eva's magnificent talk, remarkable concluding remarks and general discussion? Maybe two comments to come back to Isabelle Mensui's sentence. How nice it is to have such an unconventional discussion in this place, which is associated with discovery, but also some tradition. You have been talking against dogma, but now we have reached a stage where this discussion is not anymore unconventional. It's a new field of research, and I'm very hopeful that in the near future we shall have the opportunity to have a new meeting on this subject. And the second comment was by Eva was, should we remove the question mark out of the title of this meeting? Personally, I'm not an expert in the topic, in the subject, but I am tempted to do it. Of course, noting that there is really very much more work to do, particularly in mammals, where the situation is complex, and even more in humans. And we need proofs of principle in both directions, as was mentioned in several talks, both showing a correlation between the epigenetic marks and various phenotypes, but also how inducing or removing these marks in a way or another may affect the phenotypes. All this will be done, I'm sure, is being done, in fact. So to finish, I would like to thank, of course, the speakers. I think, as far as I'm concerned, I know, talking to other people, that the whole day has been really remarkable and fascinating for all of us. I would like to thank the organizing committee. I can tell you that the ideas were not always convergent at the beginning. We had an interesting discussion how to compose the program of this meeting. It went fine. You saw it today. So Geneviève Algozni, Edith HEARD, François CLUZIN have been very instrumental in organizing, putting together the program. And I would also like to thank Sandrine Chermier and her group for the logistic of this meeting, and our president for nice work that he did the whole day.
Thank you very much. Thank you, everybody. It was a very nice and fruitful session. Thank you.